DNA, or deoxyribonucleic acid, is found within the nucleus of every cell of every living organism. DNA is often compared to a blueprint because it basically contains all the instructions on how a cell will grow.
So DNA molecules are made up of nucleotides. A nucleotide is made up of a phosphate, a sugar, and a nitrogen-containing base. A DNA molecule is composed of two chains of polynucleotides. On each chain, the sugars and the phosphates link together to form a backbone, hence the name sugar-phosphate backbone. These chains run in opposite directions. One goes up and the other goes down. That's called antiparallel. Hydrogen bonds between the bases of nucleotides hold the two chains together. There are four distinct nucleotide bases: adenine, thymine, guanine, and cytosine. The complementary base pair rule states that adenine can only bond with thymine, and guanine can only bond with cytosine.
DNA is often represented in books and diagrams in the form of a ladder model. The sides of the ladder represent the sugar and phosphate backbone, while the steps represent paired nucleotides, bases held together by hydrogen bonds. Now, while this model is useful in mapping out the parts of a DNA strand, it does not depict the actual shape of a DNA strand. The famous double helix is DNA's actual three-dimensional shape.
So now that we've covered the basic structure of DNA, let's find out how the information stored in a strand of DNA creates a protein. Proteins are chains of amino acids held together by peptide bonds. A gene is a sequence of DNA that contains the information to build one protein.
There are two types of proteins: structural proteins and functional proteins. Structural proteins form physical structural parts like keratin, which makes up your hair and nails. Functional proteins are proteins that perform actions, for example, hormones and enzymes, such as these two right here.
We begin our journey in the nucleus of the cell with the process called transcription. To demonstrate this, let's pretend that the gene sequence ATTGCCGTACC creates a structural protein. So the DNA message to create a protein comes in the form of RNA.
RNA, just like DNA, is a nucleic acid. And just like DNA, RNA has 4 nitrogen bases. But instead of having thymine, RNA has uracil.
So transcription is basically transcribing the DNA sequence into messenger RNA. However, there are some rules to follow when transcribing the DNA into RNA.
Just like in DNA, certain base pairs go together. A T in the DNA sequence would become an A in mRNA, and an A in DNA would become a U in mRNA. Guanine and cytosine would still go together.
With this knowledge, we can now transcribe our DNA sequence into mRNA. ATTGCCGTTACC would turn out to be UAACGGAAUGG.
The mRNA sequence leaves the nucleus as sent to the ribosome. The ribosome then reads the message and produces a protein matching the specifications laid out in the gene.
Now, it will be hard to show protein folding with such a short sequence. Now let's imagine that the resulting protein ends up looking like this.
But proteins are not long linear chains. They fold up, and there are rules for this protein folding. Interactions between amino acids cause the folding and bending of the protein. The amino acid sequence of a protein determines its shape, and the shape of a protein determines its function. So let's use this legend to determine how the protein will fold and its resulting shape. Point of note, these graphics are simplified. And now we have our beautiful structural protein.
However, proteins do sometimes fold improperly. There are a few things that can cause protein folding to go wrong. For example, a person may possess a mutation that changes a specific amino acid in the protein chain, resulting in the protein folding in the wrong manner, such as this clump right here.
There are several diseases that are known to be the result of improper protein folding. Loss of function is a type of problem that occurs when a specific type of protein fails to fold properly, resulting in a shortage of that specific protein required to do a specific task. Cystic fibrosis is one such example.